you to the organizers for inviting me to present some of my PhD projects. Okay, so I would like to first thank my supervisors, my PhD supervisors, Henri and Vincent Calvez, for guidance and supervision, and Sarah Otto for allowing me to visit her lab for almost a year. And the project was actually initiated with her in particular, and so I would like to acknowledge that it was initiated on the traditional and unceded land from the Squamish and Musqueam First Nations. The motivation comes from if you consider a species in heterogeneous environments, two patches linked by migration, back and forth migration, and you have selection that is locally stabilizing towards a certain optima that are different in the two dims, and you consider the adaptation with a quantitative trait, here the color. Then you can expect that there's a large variety of genetic architectures that arise from this kind of local adaptation. And in particular, there's a simulation study that shows that you can have very strong response to certain migration levels. And so the objective that we had is to kind of give a theoretical framework in order to study the local adaptation of large sexual populations to this kind of patch environment with hybrid genetic architecture, where you have major responses coupled with a lot of polygenic kind of quantitative contribution. So the migration selection models are not new at all. There's a large number in population genetic models that looks at discrete traits like eye color. I just put a few, but mainly they either look at a few loci, or there's some with a lot of polygenic traits and with a high number of loci, but with equal effects, to my knowledge. And on the other side of the spectrum, you have quantitative genetic models that look at more continuous traits. You can distinguish sexual and asexual reproducing populations. In sexual population, you have kind of a Gaussian approximation of local trait distribution analysis. I would like to point out that in this simulation study of Deba, Iman, and Guillaume, the authors explore numerically how large effect can skew your population away from Gaussian distributions. And in asexual population, where you have mainly a continuous traits labels. And as for hybrid models, in terms of hybrid genetic architectures models, there exists in particular to one particular problem, which is if you have a sudden change in environment, a sudden optimum change, then will you either sweep a major response, or would you have like a shift in all the frequencies of polygenic traits to follow the optimum? And so a first study from Lundy in 1983 is kind of looking at major genotypes and put a Gaussian noise around this. And that's interesting, and it relates to some of the works we did. Precisely, the genetic architecture I would like to consider is the following. So you have hybrid traits, which is the sum of a major effect. You have plus or minus eta corresponding to big A and little a here, and a quantitative background, which is the sum of many loci that are the allelic. And here you have a parameter, which is the variance at linkage equilibrium of the quantitative background, which means if you just distribute all the loci randomly in your population and you can look at the variance of one genome, then this is what you will get. As I'm looking at large population, I'm mainly looking at a PD framework where I have 4 PDEs that are coupled. And so you follow Ni, so 2 DIMs and 2 allelic densities that are denoted by big A and little a. And these are subjected to reproduction with a certain operator B, A and little a, with a reproduction lambda, a quadratic selection towards an optimum. And the trait under selection is the sum of the quantitative background Z and the big allele effect eta. You also have a non-local competition, so it's a nonlinear, non-local PD, and you have migration that couples the two dims, so you have J and I here. Okay, so I still haven't really talked about what is behind these reproduction operators, and so I'm going to do that now. So imagine if you have very many loci that are the allelic, then if you have two parents, which are these very cute snails, then one offspring that might arise is kind of this one, where the genes are mixed with the parents, between the parents. And this is kind of the idea between the model that look at within family trait distribution, and that kind of describes it as a Gaussian distribution of offspring in the spirit of a central limit theorem that was really precise in this paper in 2017. And so we look at a limit in large population where inbreeding doesn't really erode that segregational variance, that is the variance of the offspring, and we will take it as a parameter and constant across families. And it's actually, yeah, we can link it to the variance of linkage equilibrium, but we'll keep sigma square for simplicity. And so in the view of partial differential equations, you end up with that kind of integral operator, which is non-local because an offspring can be both far from both parents, and it's quadratic because you have two parents, you have to multiply the densities. So an offspring is born with a trait Z that is deviating from a Gaussian distribution from the mean parental traits Z1 plus Z2 over 2. Okay, but now if you have, in addition, a major effect, then if you have two parents with different alleles, big alleles, then your offspring can have either of these alleles. And so what you end up having is first you choose one or the other allele, and then you have a Gaussian distribution of the quantitative background around this effect. And so you get a bit more complicated reproduction operator, and it's quite similar to the one we had before, but you can notice that you have actually two integrals, because if you're born with an allele A, then it means that one of your parents must have an allele A, so either both have them or just one. So that means that both have them, or one has big A and one has little a. You notice that if you lose, in the population, if you lose one allele, then you come back to the infinitesimal model previously defined. Okay, so we have this PD model, and we're going to look at it in a certain regime where the variance of the quantitative background is small compared to how the habitat are heterogeneous, or also how the major alleles diverge, that makes the trait diverge. So essentially, it's a small quantitative contribution. And so because of that, you need to look at large time scales, and you introduce a small parameter, epsilon, which is the ratio that you want it to be small. But you also have to rescale the time accordingly to kind of see the effect of the quantitative background. But so you obtain these non-bolded PDEs. Okay. And you can notice that if you lose an allele, then you come back to a model with only two PDEs, you lose, so you have fixed allele. And the steady states have been, were studied in a paper that I published. Okay, so what that means is that what we really want to know is when is the major polymorphism still viable in the regime that you look at, which is epsilon square very small. That is an illustration of such polymorphism. So you have each color corresponds to one habitat, and the dashed lines are for big A and the dotted are for little a. So each allele is favored in one of the environment because it helps you adapt to that environment. And so it's highly non-Gaussian, for example. Okay, so it was a bit ambitious, so I'm going to try to do everything. If I don't, this is the strategy of the whole analysis. So we start from a PD model, and we're going to use some convex analysis arguments to close an ODE system around the moment of the distributions. And from there, we're going to let epsilon vanish and show that there's a separation of time scale that arise. And we will study the stability of the major polymorphism in the limit system. Okay, so the first presentation talked about the Hamilton-Jacobi framework. It's kind of linked to what I will talk about. So the small variance methodology was introduced by the paper that was cited before for quantitative genetic models. And it says that if you have small diversity, quantitative diversity introduced by reproduction, and compared to how selection concentrates, then you will concentrate on the fittest trait. And then the fittest trait will consider as direct singularities, and direct singularities are not nice to analyze, so you want to define something that will kind of unfold that. And what I mean, that is a bit cryptic, so that's an illustration of what it is. So if you take a spiky gray distribution, which is a Gaussian that have a very, very small variance, then if you look at the arguments in the exponential, that is a quadratic, and that is smooth, and so you can do like Taylor expansion on it, and that's the idea. So you're gonna look at this quadratic curve that hopefully retains some information, some fundamental information on how you adapt, for example, the value of the fittest trait. So an example of how you use that in homogeneous space without any major effect is, okay, so if that ansatz is smooth, then you can do a Taylor expansions. Well, let's say we can. And what you are interested first in is the main term, which will define mainly your profile. And the first thing that you can notice is that if you want your population not to go extinct or to blow up, then because you have U naught over epsilon square, U naught must be non-negative, and it must reach a maximum in 0. Otherwise, it would go to 0, the population size. Okay, so it's non-negative, it can solve on an empty null set. And it's a null set because if you have an interval, then it will blow up. Okay, so let's say we have homogeneous space where just reproduction, competition, selection. Then you divide by an epsilon, and you got bounded quantities. And imagine that you look at the stationary state, then you kind of want to know what happens to that ugly reproduction term when epsilon is vanishing. And if you explicitly compute that, so that's your infinitesimal model. So you have the Gaussian deviation from the mean parental traits, and you plug your ansatz in it, so your U0, your U0, then you have formally that is bounded, the corrector is bounded, but formally you have here at the numerator something that must remain negative, otherwise same kind of thing. You divide by epsilon squared, you take the exponential, that would explode. So it gives you a very strong constraint on u0 to verify that property for all z, which is the maximum of a certain quantity, is zero. So the maximum of a certain quantity equals 0, but actually that determines completely u0 as a quadratic function through some convex analysis arguments. And if you have that argument, then plugging u0 here would yield that your distribution is Gaussian at the main order. And I'm just going to detail a tiny bit what kind of arguments there is. And it relies mostly on a convex conjugate object, so defined like that. So the important part is that from the constraint here, to which you want to find the solutions, then it defines a functional equation on the convex conjugate. Okay, and this is a not so hard functional equation, because if you iterate that, so you take the arguments and you divide by 2 each time. So if you iterate that a lot, then you can actually show that it's quadratic. And taking the biconjugates, which is the convex envelope of u0, then you can show that u0 is quadratic. So that is the main point. But in our case, since we have a cross term between these major alleles, we have a constraint that is a bit more involved, in which that you don't have the maximum of something that is equal to 0, you have the maximum of the maximum of two things that are equal to 0, because you have a sum of Gaussian, sum of integrals. And that is a lot weaker a priori, because if you have maximum of two things equals to 0, it doesn't mean that any of them are equals to 0. Okay, and the point is you introduce the convex conjugate, and it verifies a new functional equation that is a bit more involved, because it has a maximum of two quantities. Okay, that is not the same as before. But if you take the maximum of the two, then the maximum actually verifies the same functional equation as the previous case, and so you can actually say that it's y squared over 2, and if you take the minimum, you can show that it's lower than the maximum, and you can show that it's also y squared over 2, and point is that both your main terms are quadratic and centered at the same point. And that means that your allelic local distributions, so per allele, per dim, are Gaussian at the main order. And so you can go from the PD model and just integrate to look at the moments, because your Gaussian, the skew was going to be 0, the kurtosis is going to be like variance. So you can go reduce to an ODE system. And the small caveat is that we haven't actually controlled these corrector terms, and that is probably very technical to do, because it's really technical to do it even without major locus or in homogeneous space. But okay, so the next part, I'm sorry, that was a bit heavy, but the next part shouldn't be too much. And so the idea is that we have an ODE system, and we want to do a it has a slow, fast analysis framework, in the sense that if you have the slow evolution variable in the x-axis and the fast ecological in the y-axis, if you start not here, you will very fast adapt to a certain ecological level, which is an equilibrium corresponding to where your evolutionary variable is. And when you're on that manifold, that is the manifold of the ecological equilibrium, you will go to a global equilibrium of your system moving on that manifold. We had an ODE system with 8 equations, and between them, we can show that there's only one that is fast, which is the mean quantitative background between all the alleles and all the dims. And so you have 7 fast, which means that between all the mean traits, the mean quantitative background, the only slow information is the mean of them, the average between them. And so you get a system of equation where you have explicitly a fast time derivative here, and here you have a slow dynamic. And at the limit, you can show the convergence to an algebraic system and a slow dynamic on the manifold that is defined by this the fast ecological equilibria. So the system is also always at ecological equilibria, and the evolutionary variable moves. Okay, and to show that convergence, the critical point is actually to show that the arrows are really that strong, and so it's the stability of the slow manifold which is defined. So G is in a 7th dimension space, so it should be quite hard, but it's actually surprisingly not, because it has a very particular algebraic structure that is separated by blocks. And so you can have an explicit description of, so if you fix a z, you can find an explicit solution for all the ecological variable. I put an exclamation mark because actually when you don't have the major locus, then you can have many more, can have up to 3 solutions, and it's not explicit at all. So adding variables and adding equations, in that case, adds a lot more constraint, and you have only one solution. And the crucial argument for the convergence to these single dynamics on a slow manifold is the local stability of that manifold. And you do that by Roth-Hurwitz criterion, but it's very miraculous because it's highly nonlinear. There's quadratic terms everywhere, and it's like a big matrix, but it works. And it's very robust. It's actually written for asymmetrical environments, and I believe it's also robust to have more general selection function than quadratic ones. And so the next part is actually to look at, we wanted to know what was which condition the major polymorphism big A, little a could be stable. And that's looking at the slow dynamic, where it's going to go to. And that is, in a nutshell, the result. So if you have the selection that is increasing y in the x-axis and the migration rate in the y-axis, then the polymorphism is stable only in a kind of narrow part, and it's unstable at low selection, so here, and it's unstable too at high selection, which is kind of surprising, because you would think that high selection is favorable for this dimorphism to happen, because it helps you locally adapt to one of the 2 dims. And actually, it is really due to this quantitative background, because that is the upper bound of stability when you don't have the quantitative background, when you just have one locus that is giving you the local adaptation. And so the red crosses are the parameters values for individual-based simulation that I took to confirm that, because it was, well, frankly, very surprising. So that is kind of the framework. We have discrete generation with small length, so that it can discretize well the continuous time model. We have very big populations. We take a bunch of alleles, and that are the, so at weak selection, so that is what I plot is the variance at the major locus. So, and if it reaches 0, that means that you lose one allele. And the dashed lines are the continuous trajectory, the continuous time PD discretized trajectories, and the field one, or the individual-based simulations. So that at weak selection, you expect migration to really mix things up and lose your major polymorphism. At intermediate selection, you keep 2 alleles because the divergence at the major locus kind of helps you adapt. But at strong selection, you lose both alleles quite quickly. And the 2 models are in agreement on that. And my interpretation is that at strong selection, you have like a concentration effect of the quantitative background that kind of binds so you want to adapt divergently, but your quantitative background kind of wants to pull you like that, and it wins. It helps that nothing can mutate also. And so if you lose your polymorphism, then you get to the monomorphic state, and theoretically, all the other outcomes have been established. So that is the 4 pictures that you got, but maybe I'm a bit over the time. As a conclusion, can I just conclude? Yeah, sorry. As a conclusion, we also propose a toolbox to describe how we like the method to apply it to more complex population genetic models in like with very many dames, if you want, very many habitats. So the caveat is that the major alleles cannot mutate in our model because it's mathematically hard to combine segregation between all the small loci and a diffusion in the major alleles space. But that is my next objective regarding this. And so I don't know if it was the best of both worlds, but yeah, I mean, I can't compare. Okay, thank you.
Thank you very much. Questions?
So in the strong selection regime where you cannot maintain the polymorphism at the main locus, do you still have local adaptation due to the quantitative trait loci? Or
no.
Okay.
No. So it's actually a regime where when you lose the major polymorphism, you will go to a monomorphic state because there's too much bindings of the segregation. And you will go to a source sink scenario where you're adapted to mainly one habitat and you send newcomers to die in the other. But it's really it's highly related to that special regime in which we are, in which the variance at linkage equilibrium of the quantitative background is small compared to the heterogeneity of the habitats. And it will not happen if it's not the case, by the way.
More questions? Well, I have one. So any chance of having like an experimental way of seeing that there is an allele linked to the... because you have like one strong allele and then the infinitesimal model. So if you can identify the small allele effects, is there a way to see that actually there should be like another one to look for?
So, the debate between like major responses and polygenic responses is a huge one in evolutionary biology, and there's experiment that shows both responses in different condition in the field, in a lab, it's different. In terms of identifying, so I'm not very used to statistical models to actually GWAS, genome-wide association studies to kind of see that. I imagine that it's quite hard to actually have these tiny alleles because they are tiny, and I imagine that I don't really know. That's very theoretical, and I think a very important question is what is the balance between the mutation at the major effects versus what is the variance of the small variance? And that's, I think, because if it mutates, you kind of expect the major allele to just mutate until it reaches the optimums. So, anyways.
More questions? Well, if not, thanks all the speakers of this morning. Yeah.